Hi everyone, welcome back to another video, and today we're kind of dialing back to talk about the fundamentals of biochemistry, because I know I very quickly went into some very complicated pathways, and sometimes I forget that it's been a while since I've actually studied the fundamentals of biochemistry, and that it might make a little bit more sense to cover some of these details before moving on to some more complicated pathways. So today we're talking about just kind of how enzymatic reactions work, how they happen, why they happen the way that they do, because if you understand this, then it's going to be a lot easier to understand what happens when you remove enzymes in other processes. And so let's just get into this. So we have right here kind of the most simplified form of chemical reaction. We have A, which is a compound. This is called our substrate. This is what we are starting with. And then A is going to be converted, in this case, to C, which is our product. And then this is going to happen by an enzyme B. And the enzyme is a catalyst, so it is going to make the reaction happen in a more rapid time frame, because a lot of times you might have A that would spontaneously convert to C, but that could happen over a long period of time, and the probability is very low that it would happen by itself. So we add an enzyme B, which is going to make this happen within a reasonable time frame, to make it actually relevant to bodily processes and have it be a stable method of producing what we need to produce out of it. So I can make this hopefully make a little more sense. We're going to draw out some diagrams to kind of explain this process we have here. So let's imagine we have a lot of A. A is just floating around somewhere in the body or in a test tube, wherever this reaction is happening. And we have a bunch of A's. And then we have a bunch of B's. And B, we're just going to draw this little Pac-Man here. This is enzyme B. They're not all going to look good, but that's fine. You get the idea that we have enzyme B. And so what's going to need to happen, notice we have 6 A's and we have 3 B's. And so if you notice, it requires B to get from A to C. And since we only have 3 B's, that means only 3 A's can bind. So these A's are going to bind to B, and they're going to form these little complexes. So let's say we have, this looks really stupid, but I hope it gets the idea across. So we have 3 of these now that are A bound to B. And then if A is going to be converted to C, then over here, we're going to have an end result of 3 C's. Okay, so even though we had 6 A's, we only have 3 of enzyme B. So then those 3 B's get filled up by A, and then those A's are then released from B and converted to C. And then once that happens, then these 3 B's can be empty once again and take another 3 A's. And so if you look at this, this is something that we call our limiting reagent. And so this means that it doesn't matter how much of A we have. We can only ever make 3 C's because we only have 3 B's. But let's say if we did this a different way. So let's say if A was the limiting reagent, if I just erase all these. But let's pretend that A is our limiting reagent. And let's say we have a lot of enzyme B. So we're going to have so we have 3 B's still, but we only have 2 A's. Okay, so even though we have 3 B's, only 2 of these A's can bind. So we're only ever going to produce 2 C's. Does that make sense? So there's always going to be one of these items is going to be less than another one. And whatever we have the least amount of is going to determine how quickly these reactions can happen. Because sure, even if we have more B's, or we have more A's show up and we have fewer B's, then we can still convert all of the A's. It's just going to take more rounds. Like it's going to take if one batch is 3 and we have 6 A's, we can still get through all 6 of the A's to convert them to C, but it's going to take multiple rounds of these enzymes. Whereas if you increase the amount of enzymes, then you can do it in a shorter amount of time. And so this is why we have this change in concentration and ending up favoring this product or favoring a substrate. So if you have a reaction that wants to go from A to C, and we have a lot of A's, and we have enough B to convert all of the A to C, the reaction is going to have a high probability of being able to move in this direction because we have enough A to bind to B to be converted to C. All right, but back to our other example, if we don't have a lot of A, but we have a lot of C already, let's say that we already converted a lot of our A, and so we only have 2 A's left, but we're all done with our A's, and we've made a lot of compound C. This is going to slow down because we're running out of A. We don't have enough A to make this reaction go forward. So even though we have enough B to produce C, we don't have enough of our starting product. Like, let's say if you're making cookies, all right, and you have some cookie dough, and then our oven is our enzyme. Our oven is going to take our cookie dough and convert it into a fully baked cookie. So if our oven has enough space in it, but we don't have enough cookie dough, and we have a lot of cookies, we can't make any more cookies because we're out of cookie dough. All right, so we have a low probability of being able to make the end product. And so this is how we drive our reactions. So then adding kind of another layer to this, let's say we have a lot of A. Okay, actually, you know what? We're going to add another step to this. Let's say that I should make this smaller. Let's say that this is now a 3-step process. And we have A converted to C by enzyme B, and then we're going to have product C, which is now going to be our substrate for this reaction, where C is going to become D, and this is going to be converted by enzyme E. All right? So let's draw this out. So we have some A, and then we have enzyme B, which is this little moon-shaped thing. And then we have 4 A's. Only 2 of these can bind at a time. And so then we're going to produce 2 C's out of this. And now we have enzyme E, which, I don't know, let's make this little upside down moon shape. This is E. And we only have, let's have 2 E's. Just make this easy. So then we have our 2 C's that are produced can bind to enzyme E. And then we can produce 2 D's out of this. Okay? So does this kind of make sense here? We have kind of our limiting reagent here is enzyme B, because we have a lot of enzyme A. A is going to bind to B, going to be converted to C's, and then everything else down here is equal. The amount of C we're producing is equal to the amount of E we have, and so we're going to get the same amount of D in the end. But let's say, for a second, that enzyme E does not work. We have a deficiency of enzyme E. So let's draw this out and see what would happen. And this is what we're getting into with my pathways down the road. This is how we get a buildup in substrate when we take out the enzyme. And so we're going to draw this out, and I'll start erasing these. I'll leave the step A because we're not changing much of that. All right, so we have substrate A is going to bind to enzyme B. We're going to produce 2 C's. But hold on a second. We don't have E. So we can't do this step down here. This is not going to happen. So since we're producing C, so let's say this round already happens. We finish that. But then the next round, these A's are going to also need to be converted. But normally what would happen is these C's would be converted to D, and we'd leave space for enzyme E to catch up and so on. But we don't have that. So now we just have these C's that can't go anywhere. And now we have A's, which are also afterwards going to bind to enzyme B. And we're going to produce more C's. So now we have a lot of C, and we don't have anywhere for them to go. So we're going to have a really high amount of C. But then let's say again, we get more A that's being produced. We got more A in the body, but we have so much C that we are no longer favoring this reaction. We have so much C that this is now going to favor this direction. And this is something in biochemistry that we call the law of mass action. Mass action. So if we are favoring something called equilibrium, where there's equal amount of substrate and product on both sides, we're trying to reach a balance in both, but this equilibrium ends up usually not being reached because we end up converting C to D, but we don't have that. So then the product's going to build up. And so now we have more C than we have A. And so it's going to favor production of A. And then we're going to back up this A. And so at the end, we're going to start decreasing the amount of C, increasing the amount of A. Everything is going to back up. And so this is a lot of fundamental information about biochemistry. I hope I explained it decently enough. It seemed like I was kind of ranting, but it's if I can clear anything up, please let me know, and I can do some other fundamental videos in the future. But in the meantime, please leave your comments, questions down below, like this video, subscribe to my channel, and I will see you all in the next one.